Hello everyone. Today, we will begin studying the preparation of culture media.
培养基是指人工配置而成的适合微生物生长、繁殖和积累代谢产物所需要的营养物质。无论是研究微生物还是利用微生物，都必须配置适宜微生物生长的培养基。它是微生物研究和发酵生产的基础。本项目主要介绍培养基的概念和组成成分，并通过这部分知识的学习，让大家了解培养基的应用。培养不同微生物所需培养基不同，对于同种微生物而言，由于培养目的的不同，对培养基的要求也是不同的。因此，培养基种类很
according to the presence or absence and the amount of solidifying agents in the culture medium. Culture media can be divided into liquid media, solid media, and semi-solid media. According to the different sources of medium components, culture media can be divided into natural media, synthetic media, and semi-synthetic media. According to the intent use of the culture medium, they are divided into basal media, enrichment media, selective media, differential media, and production media. 1. Liquid media. Liquid culture medium is a uniform nutrient solution prepared by dissolving various nutrients in a fixed volume of water. They are liquid culture media for microorganisms or plant and animal cells. It has the advantages of supporting both aerated culture and shake culture. Under static conditions around the bacterial or cultured cells, a diffusion barrier to oxygen forms, hindering nutrient uptake under aeration or shaking conditions. This barrier can be eliminated, thereby increasing oxygen supply. Therefore, it is beneficial for cell growth and increases production yield. Liquid culture media are commonly used in large-scale industrial production, as well as in basic theoretical and applied research on microorganisms in laboratories. Different types of culture media generally contain the carbon sources required for microbial growth, in organic salts, growth factors, water, and other nutrients required for microbial growth, with appropriate proportions of each component. 2. Solid media. A solid culture medium refers to a medium that, after adding a certain solidifying agent to a liquid culture medium, is rendered into a solid state. At ordinary cultivation temperatures, culture media that exist in a solid state are all called solid media. One classification of solid media. Solid media can be divided into two categories. One category is made from natural solid materials, such as culture media prepared from potato pieces, bran rice bran, soybean cake powder, or peanut cake powder. Such media are commonly used in alcohol plants, breweries, etc. The other category is prepared by adding a solidifying agent to a liquid medium, such as the agar slant and agar plate media commonly seen in our laboratories. These media are widely used for microbial isolation, identification, preservation, enumeration, as well as observation of colony characteristics. 2 solidifying agents for solid media. The solidifying agent in solid media is generally not a nutrient for the microorganisms. It only serves a solidification function. An ideal solidifying agent should possess the following characteristics. 1. Not decomposed or utilized by the cultured microorganisms. 2. Remain solid within the temperature range for microbial growth. When cultivating thermophilic bacteria, high temperature tends to liquefy the medium. This problem is usually solved by appropriately increasing the solidifying agent content. 3. The solidification point should not be too low. Otherwise, it is unfavorable for microbial growth. 4. Non-toxic to the cultured microorganisms. 5. Stable during sterilization and not destroyed. 6. Good transparency and strong adhesion. 7. Easy to prepare and low cost. Commonly used solidifying agents are agar, gelatin, and the silica gel. The table below lists some main characteristics of agar and gelatin. 3. Semi-solid medium. A semi-solid medium is prepared by adding a small amount of solidifying agent, for example, 0.2% to 0.7% agar, to a liquid culture medium. This medium is often used to observe bacterial motility, isolate anaerobic bacteria, and identify strains. Semi-solid agar medium is mainly composed of peptone, beef extract powder, sodium chloride, agar, and other components. Peptone and beef extract provide nitrogen sources, vitamins, and minerals. Sodium chloride maintains balanced osmotic pressure. A small amount of agar acts as the solidifying agent. For seed medium, a seed medium is specifically used for spore germination, massive growth, and reproduction of microorganisms to produce sufficient biomass. It generally refers to media for seed tanks and shake flask seeds. Its function is to obtain sufficient, high-quality, and robust microbial cells. Precautions for preparing seed media. 1. Nutrients should be abundant and complete, including easily utilizable carbon sources, nitrogen sources, inorganic salts, and vitamins. The content of nitrogen sources and vitamins should be relatively high. Nitrogen sources usually include both organic and inorganic nitrogen, because amino acids in natural organic nitrogen sources stimulate spore germination. Inorganic nitrogen sources are beneficial for mycelial growth. 2. The composition of the medium. The medium composition can maintain pH within a certain range to ensure enzyme activity during cell growth. 3. The total nutrient concentration should be slightly dilute to maintain a suitable dissolved oxygen level, favoring massive cell growth and reproduction. 4. The nutrient composition of the final stage seed medium should be as close as possible to that of the fermentation medium, so that the seeds can quickly adapt and grow rapidly after being transferred to the fermentation medium. Inoculum volume varies with different media and microbial strains. If no relevant instructions are available, the gradient inoculation method can be used to determine optimal medium conditions. The specific operation is inoculate the strain at gradient concentrations. Culture, compare results, and use the concentration meeting expectations as the final condition. 5. Fermentation medium. Fermentation medium is a medium used for microbial accumulation of large amounts of metabolites using microorganisms. A fermentation medium is not the optimal growth medium for microorganisms. It is suitable for strain growth, reproduction, and product synthesis, in addition to enabling rapid growth of seeds to a certain mycelial concentration after transfer. It must also allow the cells to quickly synthesize the target product. Fermentation media are characterized by high total nutrients and a high proportion of carbon sources. They also contain specific elements, precursors, accelerators, and inhibitors required for product formation in industrial fermentation. Fermentation media must meet the requirements for fermentation performance and microbial fermentation control. Nutritional requirements for fermentation. Media 1. Nutrients should be appropriately abundant and complete, favoring mycelial growth without overgrowth that inhibits target product synthesis. 2. The medium has stable pH maintained within the optimal range for target product synthesis, according to the biosynthetic characteristics of the target product. Add specific elements, precursors, inducers, and accelerators beneficial to product synthesis. 4. Control raw material quality to avoid production fluctuations caused by raw material variation. This is the end of today's lesson. Have you learned it?